Hello doctors, today we will learn all about the glycolysis. Each enzyme, each step by step we will discuss. First we will see about the definition. Breakdown of the glucose to pyruvate or lactate is for energy is process is called as the glycolysis. Glucose to pyruvate in aerobic condition and glucose to lactate in anaerobic condition. Next, see the types of glycolysis: aerobic and anaerobic. Occurs when oxygen is plenty. Final product is pyruvate with 7 molecules of ATP. And in anaerobic glycolysis occurs when oxygen is less. Final product is lactate along with production of 2 ATP. Then see the location of the glycolysis. In the tissue, all the tissues of the body, it's only pathway that is taking place in all cells of the body. And in intracellular side, there is a cytosol. Then the starting material is glucose and end product is in aerobic pyruvate, in anaerobic lactate. Next we will see about the conditions for aerobic glycolysis. In tissues with low oxygen, inactively contracting skeletal muscles, and in cells like red blood cells and the lungs which do not have mitochondria. Next we will see the exact glycolysis pathway. It occurs in 10 enzymatic reactions catalyzed by various enzymes. Reactions 1, 3, 10 are irreversible and rate limiting steps. All other reactions are reversible steps. The overall reactions can be represented as glucose, pyruvate or lactate plus ATP. First phase is the energy requiring phase or preparatory phase, and second phase is energy generating phase. Let's see, these are the phases of glycolytic pathway. First phase, energy requiring or preparative phase, that is from glucose to fructose 1,6-bis phosphate, and then it conversion to glyceryl 3-phosphate. 1 to 5 steps, here is no ATP generated. And in second step, energy generating phase, to glyceryl 3-phosphate to finally to pyruvates. We will see them step by step. Here is the first step. Glucose is phosphorylated by ATP to form glucose 6-phosphate catalyzed by the hexokinase or glucokinase. It is the irreversible step. First rate limiting step of the glycolysis, and these are the isomers with different Km values. It's not that much important for exams, you can remember. And glucokinase regulated by insulin, hexokinase is not regulated by insulin. This is the step. Glucose is converted into glucose 6-phosphate, requires ATP, and these enzymes, if liver then glucokinase, if in muscles hexokinase. Activation of glucose and utilization in various pathways. Glucose to glucose 6-phosphate, then it's converted into undergoes into glycolysis or glycogenesis or lactose synthesis or HMP pathway or uronic acid pathway. Only after activation it can be metabolized further by various pathways in the cells. Glucose 6-phosphate is a central metabolic pathway. Intermediate in liver, it can enter glycolysis, glycogenesis and pentose phosphate pathway. Then the difference between glucokinase and hexokinase, it is not that much important. It's just for kind information you can overlook. Next we will see about the second step. Glucose 6-phosphate is isomerized to fructose 6-phosphate by the enzyme phosphohexose isomerase. This is the step. It is the reversible reaction, and it is the conversion of aldose to ketose. Then the third step, fructose 6 phosphate is irreversibly phosphorylated to fructose 1,6 bisphosphate by the enzyme phosphofructokinase first in the presence of ATP. It is the second rate limiting and committing step. Citrate and fluorocitrate are the allosteric inhibitors of this enzyme phosphofructokinase first. It requires ATP and second rate limiting reaction. It is the third step. Next we will see the fourth step. Fructose 1,6 bisphosphate undergoes aldose cleavage at the C3C4 bond forming two triose compounds DHAP and glyc 3-phosphate through the enzyme aldolase in the reversible reaction. Fructose 1,6 bisphosphate converts into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate using enzyme aldolase. Next step is fifth step, that is the dihydroxyacetone phosphate is isomerized to glyceraldehyde 3-phosphate by the action of enzyme phosphotriose isomerase. Thus glucose is now cleaved to two molecules of glyceraldehyde 3-phosphate, and this dihydroxyacetone phosphate is used for synthesis of triglycerides in adipose tissue, synthesis of plasmalogens, and synthesis of platelet activation factor. It is just for information. If there is a phosphotriose isomerase enzyme deficiency, there can be severe hemolytic anemia, neurodegeneration. It's the rare condition but can be lethal. This is the reaction. Dihydroxyacetone phosphate converted into glyceraldehyde 3-phosphate, and the enzyme is phosphotriose isomerase. In the second phase of glycolysis, chemical energy of the glucose is stored as ATP and NADH. Next is sixth step, undergoes the phosphorylation and oxidative dehydrogenation, and this enzyme requires NAD as a coenzyme. These steps uses inorganic phosphate to convert glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and NAD gets reduced into NADH plus H plus, and this NADH plus H plus enters in the ETC cycle to produce 2.5 NTP. This reaction, glyceraldehyde 3-phosphate converted into 1,3-bisphosphoglycerate using 2 NAD plus, it's converted into 2 NADH plus H plus, and this is the enzyme, and here is the star point that iodoacetate is the inhibitor of this step. This process of forming ATP from reducing equivalents through the ETC cycle is called as oxidative phosphorylation. This enzyme inhibited by iodoacetate. 2,3-bisphosphoglycerate is including in the Rappaport labyrinth cycle, which helps in the delivery of O2 by the hemoglobin to the tissue. In the next video, we will see Rappaport labyrinth cycle. Here seventh step, it is the reversible reaction, and it is the substrate level phosphorylation. Enzyme inhibited by arsenate, and enzyme is the phosphoglycerate kinase. 1,3-bisphosphoglycerate converted into 3-phosphoglycerate, formation of 2 ATP, and enzyme is phosphoglycerate kinase. Kinase, remember the formation of ATP. ATP is formed directly by the high energy substrate without entering the electron transport chain. This process is called as the substrate level phosphorylation. And the synthesis of ATP utilizing metabolic intermediates does not require ETC. It is the important step, and these are some examples for substrate level phosphorylation. Next, eighth step, by the shifting of phosphate group from the third to second carbon atom, enzyme and Phosphoglycerate mutase is the enzyme. 2-phosphoglycerate, two molecules of 3-phosphoglycerate converted into two molecules of 2-phosphoglycerate, and the enzyme is phosphoglycerate mutase. It is a change only 3 and 2. Next is the ninth step, high energy compound by the action of enzyme enolase. Mg plus 2 is required and activity of this enzyme is irreversibly inhibited by the fluoride ions. It is the application of this enzyme that often in large hospitals laboratory there is a time lag for few hours between the sample collection and analysis. To prevent blood glucose level from dropping, glycolysis is stopped and by adding sodium fluoride and anticoagulant to the sample. It is the application of this step. This is the step. 2-phosphoglycerate converted into 2-phosphoenol pyruvate by the enzyme enolase and dehydration is occur. 10th step, finally the pyruvate kinase converts phosphoenol pyruvate to pyruvate, transferring the high energy phosphate to ADP to form ATP. And formation of ATP by high energy substrate phosphoenol pyruvate at the substrate level without entering into ETC is the second substrate level phosphorylation. First substrate level phosphorylation in the 7th step and second in the 10th step. It is the third rate limiting committing step 1, 3 and 10, and it is the third rate limiting step first, second and third. This is the step phosphoenol pyruvate converted into 2-pyruvate by the enzyme pyruvate kinase formation of 2-ATP. Under the aerobic conditions, pyruvate enters the mitochondria converted into acetyl-CoA, which is then broken down into CO2 in the TCA cycle. Then the final products of the aerobic glycolysis, that is the glucose 6 carbon to form 2 pyruvate of the 3 carbons plus NADH plus H plus. In anaerobic glycolysis, glucose form 2 pyruvates, no NADH H plus. In the aerobic condition, pyruvate converted to lactate using the NADH plus H plus by enzyme LDH, lactate dehydrogenase, and the NAD form is used to convert glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, allowing it continued oxygen and release ATP. NADH plus H plus won't enter the ETC to form ATP under anaerobic conditions, resulting in net gain of 2 ATP. Then the energetics of the glycolysis in aerobic condition and anaerobic condition. Here's in the first step loss of 1 ATP, in third step use of 1 ATP, then in the 6th, 7th and 8th step formation of 5 plus 2 ATP and total net gain is the 7 ATP. In anaerobic condition is 2 ATP. The regulation of glycolysis. Glycolytic pathway has two main functions, breaking down glucose to produce ATP and providing building blocks for synthetic reactions. Glycolysis is regulated as three irreversible steps. These reactions are catalyzed by first hexokinase or glucokinase, second phosphofructokinase, and third one is pyruvate kinase. This is the reactions or enzymes of the first, third, and 10th reaction. And insulin, glucagon, and glucocorticoids are the enzyme. First one is the hexokinase. The first enzyme in the glycolysis is the inhibited by glucose 6-phosphate, a form of product inhibition, and it is also one of the rate limiting enzyme in the glycolysis. Then the phosphofructokinase. It is the crucial in glycolysis. Glucose 6-phosphate is an intermediate is used in glycogen synthesis and the HMP pathway. Phosphofructokinase is an allosteric enzyme with the citrate and ATP acting as a negative modulators, while AMP, ADP, and fructose 2,6-bisphosphate act as a modulators. Fructose phosphofructokinase second is a special enzyme catalyzes the conversion of fructose 6-phosphate to fructose 2,6-bisphosphate. You can draw this diagram for the enzyme, not require these 2 pages of theory. You can overlook for this. It's for only your mutual understanding about the glycolysis. And the third one is the pyruvate kinase enzyme, an enzyme that drives the final step of the glycolysis. It's one of the 3 key steps in this process. The enzyme's activity is regulated by phosphorylation. It's active when not phosphorylated and inactive when phosphorylated. When the high blood glucose level, glucagon decreases, that leads to dephosphorylation and inactive pyruvate kinase releases the phosphate and forms active pyruvate kinase. And when low blood glucose is there, glucagon increases, phosphorylation occurs, and active pyruvate kinase occurs, forms inactive pyruvate kinase. Then the insulin activates the 3 key glycolytic enzymes promoting the glycolysis. Fifth one is the glucagon and glucocorticoids. 3 main glycolytic enzymes are blocked by the glucagon and glucocorticoids, which stops glycolysis and promotes the gluconeogenesis. Last subheading, that is significance of the glycolysis. Primary energy production of the ATP and NADH, anaerobic energy source and precursor for biosynthesis, interconnection with other pathways like gluconeogenesis, HMP, and rapid energy delivery, and role in cancer metabolism. If you like it, like, share, and subscribe, and comment for next topic. Thank you.